Hello everyone, so here I'm recording section C, SDS polyacrylamide gel electrophoresis. So this is week 2 of what we did last week, or the week before Thanksgiving. Introduction. Last week you purified GFP by column chromatography. Now we will further purify GFP by SDS page. In this technique, proteins in your column fractions are first denatured by a detergent. Sodium dodecyl sulfate, so that's the detergent. SDS, which is negatively charged, coats the protein throughout, giving each protein a uniform charge to mass ratio, charge to mass ratio. Proteins treated in this way can be separated on the basis of size by polyacrylamide gel electrophoresis. So remember, GFP, I think it was like 37 kilodaltons. I'm not sure, based on sections A and B. So we kind of know, like, once we're purified, we should know like what size the band is supposed to be on our gel. Next, we will visualize the proteins by staining the gel with Kumasi Brilliant Blue, a dye that binds tightly to proteins. GFP is, so this dye binds to the proteins, so GFP is a protein, so it should bind to all the proteins, not to the gel. GFP is approximately 27 kilodaltons in size. Okay, 27 kilodaltons. We should be able to identify GFP based on its position in the gel relative to size marker proteins of known size. Furthermore, the results of this experiment should allow us to assess the degree of purification of GFP relative to crude lysate. So, if you remember with gel electrophoresis, okay, so we have a gel, and SDS page is a gel that we're using for proteins. So basically, remember that we use a ladder for all of our gels, and the ladder will have like known sizes of proteins. So we can say that this is like 100, and this is like 10 kilodaltons. So if this is like 30 right here, then we would expect our GFP to be around here, right? Because it's 27 kilodaltons. So if we see a band around this size, then we'll know that we were successful and we have isolated GFP. Okay, we have purified and isolated GFP. Okay, so activity C1, purification of GFP by SDS page. Procedure, obtain your samples. If you remember the first week we had our crude extract, fraction number 2 and fraction number 3 from the last week's chromatography experiment. So for those of you who weren't there, what we did last week is that we had a filter, and it was like a little white filter, and it had little beads that were hydrophobic, and GFP is hydrophobic, so we collected them. Just go ahead and reference the first video, but basically we collected the crude extract in one vial, fraction number 2 in another vial, and we collected fraction number 3 in a third vial, and this is the one that should have our GFP protein. Okay, we have to thaw and mix each sample. We have to transfer 20 microliters of the crude extract to a clean microcentrifuge tube. We add 20 microliters of protein loading buffer to this tube, and we mix well, and then we label the tube as CXB for crude extract buffer, and we store it on ice. And then we have Okay, then we're going to transfer 20 microliters of fraction number 2 to a clean microcentrifuge tube. And then we're going to add 20 microliters of protein loading buffer, so it's the same. This time we're going to label number 2B, store on ice, and then 20 microliters of fraction 3 with 20 microliters of the protein loading buffer. And then we're going to place the tube's marker on ice, prepared for use already. That's CXB number 2, number 3B, in the 65-degree dry ice bath for 5 minutes to denature the proteins. Okay. After the 5-minute incubation, immediately plunge the tubes into the ice bath to snap cool the samples. This prevents the proteins from renaturing. So if you can imagine, a protein has its three-dimensional shape, so whatever that may be. And so when you denature a protein, they'll just be like that. So we want to keep them denatured. And then we load the marker. This is our ladder, and we need 6 microliters into the second well of the gel. In the next lanes, we load 30 microliters of each sample on the gel from left to right in order. And we do the marker, and then CXB, number 2, and then number 3. The instructor will demonstrate. Turn on the power supply and run the gel for approximately 1 hour and 20 minutes, or 1 hour for 120 volts. When done, notify your TA. You will stain and then destain the gel before photographing the gel. The procedure is as follows. After your TA has separated the gel from the gel plates, place your gel in a plastic container. Add enough Kumasi Brilliant Blue stain to cover the gel. Microwave the sample for 30 seconds at full power. Note, if the Kumasi stain begins to boil before the 30 seconds is complete, remove it immediately and proceed with the next step. Turn on the orbital shaker slowly and let the gel incubate in Kumasi stain for 20 minutes. Dispose of the stain in the used stain jar. Rinse the gel with water and dispose of the water down the drain. Restain the gel for 20 minutes by adding the methanol acetic acid solution into the plastic container and agitating it in the orbital shaker. Dispose of the destained solution in the used destain jar. Just prior your TA imaging your gel, rinse the gel with DH2O and dispose of the water down the drain. Do not do this until you're ready to image. Take a photograph of the gel using the gel documentation system and place the gel in the center of the hood on the trans illuminator. Turn on your light source if you encounter problems. I usually do that for you. I take the pictures for you. Print a copy of your gel. After you have made successful prints of your gel, turn on the light and discard the gel as directed by your instructor. Okay, here is Appendix A, Protein Molecular Weight Size Standards. Biorad Range Protein Molecular Weight Marker. Okay, so then this is our ladder. And then basically it looks like the top one bands from top to bottom. Okay, so like this top one is 200. And then the bottom one is 6.5 kilodaltons. And then the next one is 14. And then the next one is 21.5. And then the next one is 31. So since GFP is 27, we expect it to be between these two lines. Okay. And so for your results, you want to make sure that you have like these, your gel, your ladder should be numbered, and then, you know, you'll have your picture of your gel, you can annotate it, and then for your results section, make sure that you write down what you're seeing. So in your results, you just say like, oh, the ladder showed all of the lines from 200 to 6.5 kilodaltons. Lane 1 contained, was this the crude extract? What was it? And then what lines are you seeing? And then what sizes are those lines? And then you move on to the next one. The following lane contained whatever extract or whatever sample, and then we observed 3 lines, and they weighed, they were 200 kilodaltons, 31 kilodaltons, and 6.5 kilodaltons. And then our last sample contained this. And remember, the results section is all facts. It's only what you're seeing. And then in your discussion, that's where you're going to have one topic sentence about the overall experiment. What did you do? What were the results? And you're going to state whether or not your hypothesis was correct. Our hypothesis was correct, or our hypothesis was incorrect. And then explain the situation. What happened? What did you see? And then why. So the discussion is where you give all the whys of what happened. So for example, if your gel came out absolutely blank, then you say, okay, it's possible that our samples were denatured because we didn't put them on ice. Like, we forgot to put them on ice for the entire lab period. Or, for that 5-minute incubation, like maybe you just forgot your samples there for like 30 minutes. So maybe everything was denatured. So it is important to, in the discussion, state everything that could have gone wrong, or if everything went right, then you just like describe the results and then what does that mean. So make sure to reference the annotated lab report that I emailed you guys in the last section, and then that document with red writing that we emailed you guys, to make sure that you have every single section for every single thing. Your intro should talk about the different techniques that you use to answer the question, which was, what was the overall purpose of this experiment? Was to extract and purify the plasmid protein, GFP. That was like our overall goal. And then what techniques did you use to accomplish that goal? So yeah. Okay, I decided to bring up the annotated lab report. So I just want to go over really quickly what are the different elements that we expect and that you will be graded on. And basically you need a title, and then you need your abstract. The abstract should be short. 1 sentence that summarizes the introduction, another sentence or 2 that summarizes your methods, another sentence or 2 that summarizes your results, and then another sentence or 2 that summarizes the discussion. And that's it. The introduction, I like 2 paragraphs. In the first paragraph, I like to have background information about like the overall concepts. So don't focus on the techniques here, just the overall concepts. So in this case, it would be protein purification, GFP. You can talk a little bit about what is the protein like, GFP. It's hydrophobic, and then just some more information about that. And then here in the second paragraph is where I like to state the overall purpose. That's how I like to start the second paragraph. The overall purpose of this experiment was to purify GFP. And then the next sentence you can say, what techniques did you use to accomplish your goal? And that could be like 2 sentences or 3 sentences describing the techniques. Remember not to add too much detail. It should be very broad, just 2 or 3 sentences. Don't treat that as a methods section, just 2 or 3 sentences. Then you give your hypothesis. I hypothesize that blah, blah, blah. What is your hypothesis? And then your last sentence should be a rationale for your hypothesis. So why do you believe that that is what is going to happen? What made you get that hypothesis or come to that hypothesis? Okay, so then after your introduction is done, your materials and methods section, but if you refer to the document that I sent out, Dr. Dulai removed the method section, so you guys just have to put one sentence that says, you know, please refer to our laboratory 002 lab manual lab 9 for this section. We followed that protocol with the following changes, and then that's where you just mention in a paragraph format the changes that you made in this section. And then for your results, you give one topic sentence. So that's a statement about the overall findings of the study. So were you able to purify GFP or not? Did you get a line at 27 kilodaltons on the gel? Were you able to or not? And then you ran 3 different samples. What were the differences between those samples? With reference to that 27 kilodalton line. So here you just give one sentence that summarizes the overall results. And then you start explaining the gel. You know, a gel was ran at 120 kilovolts for, you know, or however many kilovolts and however many minutes, and then start with the result. You know, lane 1 had 3 different lines at, you know, these sizes at 1,000 kilobase pairs and at 300 kilobase pairs. And so you describe all the lines, and then you have a picture of your gel, and you can say, you know, reference figure 1. So your gel would be figure 1, and so on and so forth. And then in your discussion, a statement showing the relationship between the hypothesis and the results. So this is just where you tell me, like, you one topic sentence, and then you say, you know, like, our hypothesis was correct or our hypothesis was not correct, and then explain about that. And then here is where you start explaining, like, what went wrong, or what went right. And then for the second paragraph in the discussion is where you would do this portion, which is a comparison of the findings of the lab with other related scientific studies. So this is where we expect you to go and find either a peer-reviewed journal or other universities that have conducted the same study or similar studies, and you reference their work, and then you tell us about, like, how, you know, what is the impact of that work, or what does it mean, or how could we improve our technique if ours didn't work but theirs did work. What can we change in our technique to maybe, like, make it work like theirs. Yeah, and then you have your conclusion. So the conclusion restates the purpose and describes what the students learned. Okay, so again, it's short. You know, the purpose of this lab was to isolate and purify GFP protein. And then it describes what you learned. So, like, your results, but, like, what do they mean? And then your literature cited. Okay, so those are all the sections for a full lab report. So make sure you include everything.